Hello students, today we're going to talk about cell transport and homeostasis. When we talk about cell transport, it's really important to understand that we're talking about how cells get things in and how they get them out. So they need things to survive to go in, and they need to get rid of waste as well. So what we call this is homeostasis. So in order for cells to maintain homeostasis, they have to control what enters and leaves the cell. So you can think of homeostasis as being maintaining an internal balance or a stability. There are different types of transport, meaning different types of ways that cells get things into and out of them. One way is passive transport, and that is the movement of materials into and out of the cell without the use of energy. Keyword, without the use of energy. Okay. Then we have active transport. That's the movement of materials into and out of the cell with the use of energy. I highlighted energy because that's really important when you look at those things. Active transport, you can kind of think of active transport as trying to move upstream, right? So if you're trying to paddle a boat, right, and you're trying to travel in a direction upstream, energy is required to do that. So that would be what we consider in a cell active transport. However, if I wanted to go downstream, passive transport would be a way to describe that. I could really just sit in the boat and kind of coast, and that's how passive transport will work. It doesn't require energy. So there are a few terms, of course, that we need to understand and learn for cell transport. So the first term you see there is diffusion. Diffusion is a type of passive transport where the molecules actually move from an area of high concentration to an area of low concentration. Now with my students, I have them place one hand high in the air and then say high concentration and move their hand down and say low concentration, or even with your voice, high concentration to low concentration. So it's really important that you remember that movement. So diffusion is from an area of high concentration. You can see in this picture here, I'm circling with my mouse, those molecules are in a high concentration. So if you can imagine, we've just taken some food coloring and dripped it into this water. They're going to start off, those molecules are going to start off in a high concentration, but eventually they're going to diffuse throughout the water to a low concentration, an area of lower concentration, this area of the water where there was no molecules previously. And they're going to do that until they basically achieve this part that we call equilibrium, but the molecules are really equally spaced. So the difference in the concentration of molecules across the distance is called what we can refer to as the concentration gradient. So the difference in concentration. So here we have a high concentration, over here in this region of the container a low concentration, and eventually we do have equal concentration. The molecular movement that occurs is a result of what we call kinetic energy. And that is the molecular collisions that result from the movement that cause the molecules to move from areas where they are highly concentrated. So these molecules up here are really, they're not just sitting there. They're actually they have energy, right? And that energy comes in the form of kinetic energy. When we talk about temperature, it is actually the average kinetic energy in a room. So if the room that you are sitting in is warm, it has a relatively high kinetic energy. So the warmer a room is, or an area is, the higher the kinetic energy. So on the other hand, if we take a room and we cool it off, we're really removing kinetic energy from that room. So the higher the kinetic energy is, the faster those molecules are going to move, because the faster those molecular collisions are actually going to occur. So molecules will eventually reach this state that is called equilibrium if they're able to move about free of other influences. So equilibrium occurs when the concentration and distribution of the molecules is the same throughout the entire space. So here you can see, again, kind of the demonstration I just talked about. You take some food coloring or dye molecules and you place a drop into the water molecules. They're going to disperse throughout those water molecules until they reach equilibrium, moving from an area of high concentration to an area of low concentration. So next thing we have to talk about is, okay, we want to know how this relates to cells. So when we talk about cells, we have to talk about the cell membrane, because that's where all of these molecules are going to get into and out of the cell. Remember, the cell membrane is the fluid mosaic model. So these molecules, these lipids, are able to move around freely. They have some cholesterol embedded that aren't pictured here. They have other molecules and proteins that are embedded that aren't pictured here. But the idea is that some things could get into this membrane and some things cannot. So the cell membrane does allow some molecules to pass freely. So those molecules will move from an area of high concentration on one side of the membrane to an area of low concentration on the other side. So examples of molecules that diffuse freely are the oxygen that we breathe and the carbon dioxide that we exhale. So over here you can see those gases, carbon dioxide and oxygen. You can see some other molecules that will move freely, like benzene. Some small polar molecules that can move in as well, water and ethanol. And then we have some large polar molecules that cannot move freely in, glucose. Your cells need glucose. We'll talk about how they get in later on, but glucose molecules are extremely important. But they cannot move freely into your cells. There has to be something there. And then we have charged molecules. In order for them to get in, they have to go in through an ion channel. And large molecules like amino acids as well. They're not going to be able to freely flow through this membrane. So when we talk about movement of molecules, especially water, we have to talk about osmosis. Now, you probably are thinking of this movie that all my students reference, Osmosis Jones. I still struggle to find the osmosis part of that movie being illustrated, but it does talk a lot about some of your immune system and immune response. So it's a pretty good movie from that perspective. All right, but passive transport, again, requires no energy. So osmosis is the diffusion of water molecules from an area of high concentration to an area of low concentration. So it's really simple. If you understand diffusion, movement of molecules from a high concentration to a low concentration, osmosis is really just a specific type of diffusion that just talks about water. That's why it's highlighted. That's why it's underlined. It's really the only difference here. All right. In some cases, those large solutes cannot dissolve across the membrane of the cell. However, water actually will. And here you can see a demonstration where we have a selectively permeable membrane, and that means it lets some things in and keeps other things out. Over here on the left hand side you have solutes represented by the purple. So over here where you have a higher concentration of solutes and a lower concentration of water, you can see what's going to happen. Water is going to move from a high concentration to a low concentration. Because there's more dissolved solutes over here, that reduces the amount of free water. So over here, all the water that was here is going to move from a high concentration to a low concentration across that selectively permeable membrane, and it's actually going to create a difference in water height in that demo. Alright, so now what we're going to do is we're going to dive into predicting the direction of osmosis. So if we have a concentration of water and a cell, we should be able to figure out what direction the water is going to move. Is it going to move into the cell or out of the cell? So a solution before we dive into that, we need to understand what a solution is, what a solvent is, and what a solute is. So a solution is composed of solute and a solvent. So the solvent is the substance that dissolves the solute. So water, if you recall, is considered the universal solvent. So it dissolves most substances, or many substances. Solute is the substance that is actually being dissolved. So if you're making Kool-Aid, water is going to dissolve the sugar and, I guess, the flavor packet as well. The flavor packet and the sugar represent the solute. The solvent is the water. So the direction of osmosis is going to depend on the concentrations of the solutions in the cell compared to their environment. So it's based upon relative concentrations with respect to the environment. Alright, so you could have cells in different environments behave differently. It really depends on what the concentration of the environment is. So let's look at some examples. So for each beaker, determine whether water will go into or out of the cell with 10% salt. So the cell itself, right here, represented by these little squiggly lines. It's kind of weird shape. Nonetheless, it's a cell, and it has 10% salt solution. So the first thing you have to remember is that water moves from a high concentration to a low concentration. Notice the key word, water. The salt is not moving across the membrane. The water is. So pay attention only to the water. Pause the video, take a few minutes, and solve these problems. Use arrows to indicate whether the water is going into the cell or out of the cell. Okay, now that you've had some time after pausing to look at this and solve these problems, you should have realized that if the cell has 10% salt, the other 90%, the other percentage of the cell is made up, in this case, we're going to assume 90% water. 90% water for this cell, so all the cells have 90% water. So up here, if the solution has 10% salt, we're assuming that the remaining molecules are just water. So that would also make this concentration for beaker A 10% salt, 90% water. So here in beaker A, you have, there's my mouse, sometimes it disappears on you. You have 90% water outside the cell and 90% of water inside the cell. Therefore, you have equal movement of water into and out of the cell. So really here you should have arrows going into the cell and out of the cell because water is going to move equally. We actually call this situation isotonic. If you look at the prefix-suffix list, iso means equal. So it is equal concentrations. Over here we have 98% water in the beaker, because we only have 2% salt, so it's 98% water. Here we have inside the cell, again, 90% water, 10% salt. The higher concentration of water is outside of the cell, so your arrow should be drawn to show that water is going into the cell. Lastly, we have 30% salt, 70% water, and again in the cell, 90% water. So water in this example is going to be moving out of the cell. So over here for beaker A, what's going to happen to the cell? Well, since since the water is moving equally into and out of the cell, it is isotonic, as mentioned before. In this situation, you have beaker B. Water is actually going to be going into the cell because there's a higher concentration of water outside of the cell. So water is going to move into the cell. That's going to cause this cell to swell up. Lastly, we have this one. We said water is going to be moving out of the cell. That's going to cause the cell to shrivel up. So if this is going to, excuse me. Okay, so looking at this, you're going to see that we have a 2% salt solution here, 98% water. So water is going to flow into the cell. Again, we call this a hypotonic environment. All right, hypo means under or less. Under or less. Well, what's under or less? It is actually referring to the salt concentration here, the salt concentration. So that salt concentration is under or less that of the cell. Therefore, we consider this a hypotonic environment. Over here, this is hypertonic, and hypertonic, we have a higher concentration of salt. So hyper actually means over or more. If you're hyper, you have over or more energy than normal. So that hypertonic environment is going to cause this cell to shrivel up. This one, an easy way to remember it, I tell my students, is if it swells up like a hippo, it's hypo. If it swells up like a hippo, it's hypo. So if you think about a hippo, they're kind of big and round and very plump, and that's exactly the way a cell will look within an environment that is hypotonic. So here are the terms. Hypertonic, higher concentration of solutes in the solution than the cell. So the cell is actually going to lose water, it's going to shrivel up. Hypotonic, lower concentrations of solutes in the solution than what are in the cell, and the cell will gain water. Next up, we have isotonic, equal concentrations of solutes inside and out of the cell, and the water is going to move equally in both directions. So how does this impact animal cells? So in a hypertonic environment, animal cells will actually experience crenation and will actually undergo shrinkage. So when we talk about hypertonic environment, this is what we call crenation, where they actually shrivel up. In a hypotonic environment, an animal cell will eventually burst and experience plasmolysis. So looking over here, this hypotonic here, this cell right here is experiencing plasmolysis. In isotonic, the cell will stay the same. So this is where our cells are happy in an isotonic environment. So animal cells, we are happy, our cells are happy in an isotonic environment. So if you're feeling thirsty, chances are pretty good that the concentration of water in some of your cells is lower. So this is very evident when you eat salty food, you get thirsty. That's your brain's way of telling you we need to get back into homeostasis. We need to get back into that balance. So how does it impact plant cells? Well, looking at plant cells in a hypertonic environment, remember that's under or less, they're going to shrivel up and they're going to become plasmolized. So that's what this situation looks like. For isotonic, remember animal cells love to be isotonic, but plant cells not so much. Plant cells actually become flaccid. So if you can imagine a piece of celery that's been left out for too long and you pick it up and it's kind of bent and really doesn't have any crunch to it, that would be a flaccid piece of celery. And then hypotonic is where plants want to be. Their cells are going to be turgid here and experiencing turgor pressure. So turgor pressure is basically pressure that's exerted on the plant cell wall. So this is a piece of celery that's going to crack when you break it. It's full of water. It's very happy. So this occurs when the plants are in the hypotonic environment, and in this state, the plant cells are considered turgid. So in a hypertonic environment, the plant will experience plasmolysis, and the plant will eventually lose turgor pressure, cause the plant to wilt. Many plants can recover from a wilt But if they go beyond that wilting point and experience cell death, then they cannot recover. But many times a little bit of water on the plant can sometimes recover them. I've seen this happen many times. You look at a plant and it looks dead, then you give it some water and it just within an hour they're just become full of water, so they become turgid again. So we've talked about some forms of passive transport in osmosis and diffusion. Next, we need to talk about other types of passive transport, like facilitated diffusion. So if you're facilitating something, you're helping, right? So again, it's a type of passive transport, does not require energy. So some of the molecules are too large or not soluble, meaning they cannot dissolve in lipids and they cannot diffuse freely across the membrane. They have to get into the cell some other way. So these molecules require a help of a specific protein in the cell. We call these proteins carrier proteins. So here's some examples of what that might look like from a cartoon version picture. So here's a protein channel and a carrier protein. So there are different types, both important in facilitated diffusion. So there are ion channels are another example of a passive transport. So the ion channels are types of proteins that basically allow specific ions into and out of the cell. So each ion channel is specific to a particular ion. So ion channels can be, as you can see over here, they can be open or they can be gated. So looking here, you can see this is an open channel and a closed one or gated. Notice right here it's gated. It has it's blocked, isn't it? The molecule cannot get through. So gated channels can open in 3 different ways. They can open if the cell membrane is stretched. They can open with electrical signals, and they can open when certain chemicals are present in the cytosol or the environment that cause them to open. So that's going to vary depending on the type of organism that you're dealing with. Next, we have active transport. So active transport is one we've mentioned before, but we haven't dove into. So all the other examples we previously talked about are all passive transport. Now we're looking at active transport, and that is the movement of material up the concentration gradient. So if you recall, that is the movement of those molecules up the concentration gradient from a low concentration to a high concentration gradient. Yeah, the high concentration. So basically paddling up the river. So it's going to require energy. So if you're trying to paddle upstream, of course it's going to require energy. So that is an easy way to remember active transport. So ion channels, carrier proteins that assist in active transport are called cell membrane pumps. So that's the difference. That word pump is going to help you identify that it is a type of active transport. The most talked about type of pump that you will hear about in any biology class is the sodium-potassium pump. And this is the example in every single book about active transport. So this is a type of cell membrane pump, and it basically transports sodium and potassium ions up the concentration gradient. So from a low concentration to a high concentration. So for some animal cells, we need to have a higher concentration of sodium outside of the cell and a higher concentration of potassium inside the cell. So by the way, Na plus represents the sodium and the K represents the potassium. So at this moment, I'd like you to pause and I want you to watch the video from Khan Academy called Sodium Potassium Pump. In that video, it's a requirement in my classroom, you go over all the ins and outs and important factors that are not necessary for me to repeat in this presentation, but please make sure you watch that to help you understand the sodium and potassium pump. So another type of movement that we are going to cover is movement of large molecules. Molecules are too large to move through the membrane or through any type of channel protein. So cells actually have a way to get materials in and out. And that is through either endocytosis, getting stuff into the cell, and exocytosis, getting stuff out of the cell. And of course, they do require energy, so they are an active transport. And it is also what we would call bulk transport. Alright, so again, endocytosis is an active transport. Endocytosis is the process where the cell is going to actually bring stuff in to the cell. So it's going to ingest large particles and external fluids, or even macromolecules. So the cell is going to basically form a vesicle around the material and then bring it into the cell. So there are two types. There's pinocytosis and phagocytosis. So for pinocytosis, transport of the solutes or the fluids. So that's the key terms there, fluids or things that are dissolved in a solution. And then phagocytosis is going to be large particles or whole cells. So the phagocytes basically ingest bacteria and viruses and then basically destroy them with the help of the lysosomes. Exocytosis is the process where the material from the vesicles are released from the cell. So that's where cells are able to get rid of their excess waste and their toxins. So these are actually packaged by the Golgi apparatus, and then they're transported to the cell membrane where they actually fuse with the cell membrane over here and are expelled from the cell. So that's exocytosis, getting rid of excess waste and toxins. And that concludes our notes on transport, cellular transport. Remember, there's a lot of key terms here. Review your notes. Make sure you watch the Khan Academy video on the sodium potassium pump, and put some extra time in studying those key terms. I hope this was helpful. Have a good day.